Hi, my name is Nina Horowitz, and I'm the CEO of ImmuneBridge. I'm here to tell you about how we're manufacturing the future of cell therapy. We are a small startup based in San Francisco, California. We have expertise across machine learning model development, bioprocess engineering, and cell therapy design. And we're backed by some really visionary investors, including NFX, Insight Partners, M Ventures, which is European Merck, and of course, Long Game. Cell therapy uses cells as living drugs to attack diseases inside the body. So the scans I'm showing you here are from a woman named Lori. She was dying from lymphoma. She had failed 6 prior lines of therapies. Nothing was working for her. And then she received a single dose of a CAR T cell. And within a month, her scans had entirely cleared. She has never received any treatment since then, and she's still healthy to this day, 7 years later. The picture on the top right is us together at the Boston Public Library just last year. Data like Lori's triggered a Gartner hype cycle for cell therapy. So people rushed into the space. They were founding companies left and right. These companies were being acquired for up to $12 billion. And then everybody realized that manufacturing cells is actually really hard, and the field contracted. And in 2025, we've been in an IPO drought. But if ImmuneBridge can solve the manufacturing challenges, we're going to usher in a new era where countless new curative therapies are being developed for all kinds of diseases. Why am I so obsessed with the manufacturing? Well, here's what it looks like right now. 20% of patients are dying while they wait for their therapy to be manufactured, and the cost per patient is between $500,000 and $1.25 million. With the ImmuneBridge platform, these numbers go to 0% and just $10,000. The other challenge with cell therapy manufacturing is finding good cells. So cells vary really significantly from one donor to the next. I'm showing you here clinical data from a trial where patients all received cells from different donors. As you can see, the patients who received cells from good donors, the blue line, had a 14 times higher likelihood of progression-free survival than the patients who received cells from suboptimal donors. So, of course, the big question here is how can we predict this in advance to make sure that every patient gets a good quality therapy? The ImmuneBridge platform solves all of these issues. So we source and screen from those donors that are actually capable of driving a cure, and then we turn them into consistent, affordable, and large-scale therapies. The way we do this is through 3 key technologies. The first is that we've developed machine learning models that can predict donor performance. The second is that we have designed a proprietary small molecule called IBR403 that lets us expand immune stem cells up to 100,000-fold in culture while maintaining their multipotency. And the third is that we've designed processes to be used in bioreactors for both this stem cell expansion and the downstream immune cell differentiation. All of these processes are GMP ready and ultra low cost. And what this means is that we're going to have both superior clinical results and superior market access because we're putting out a higher quality product at a much lower cost and a much larger scale. In terms of competitive landscape, there's really nobody who can do it the way we can. There's plenty of companies working with immune cells or induced pluripotent stem cells, but each of these comes with their own unique challenges. We are the only company capable of integrating high-throughput donor screening with stem cell expansion as a large-scale, reliable source for manufacturing. We've spent a lot of time optimizing the use of IBR403, proving its capability to develop multiple different immune cell lineages downstream, working on our engineering processes, our bioreactor processes, and now we're finally ready to start a phase 1/2 clinical trial. So we're raising $50 million, which will take us through our tech transfer, our clinical manufacturing, and the trial itself, and will also enable us to develop a bunch of additional assets through partnerships. So while internally our focus is on NK cells for cancer, we will also be developing red blood cell, neutrophil, macrophage, and even mitochondria products for rejuvenation in aging populations. And because we can make any immune cell therapy or any immune cell byproduct, we are going to guarantee ourselves revenue across diverse markets. And this is a really exciting time in the cell therapy space because it used to be that cell therapies were only used for people who were dying of cancer. But now we're seeing cells used for people with Alzheimer's disease or liver fibrosis. And who even knows what else might be possible with cells if you could manufacture them at large scale and high quality. So I want to leave you all with a quote that I love. It's from the first liver transplant doctor, Dr. Thomas Startzel. And he said, "The history of medicine tells us that what was inconceivable yesterday and barely achievable today often becomes routine tomorrow. I want cell therapies to be that routine intervention that's available for anyone who needs them, and ImmuneBridge is going to be the company that manufactures that future." Thank you so much for your time, and I'm happy to take any questions.
And congrats on your board member becoming the director of ARPA-H, which is a $2 billion a year breakthrough moonshot bio funding entity of the US government. So, yeah, with that, we love that you guys have a broad enabling technology for the entire cell therapy space. Could you elaborate on why cord blood cells are superior to, say, PBMC derived or iPSC derived?
Absolutely, yeah. So, first of all, thank you so much for your support and for giving me the opportunity. I think the important thing about using cord blood versus these other sources actually has part to do with the biology of cord blood itself and part to do with regulatory. So, on the regulatory side, cord blood comes from babies who were just born. This is actually already an FDA licensed therapeutic. These babies don't have any viruses. It's very easy to be sure that you're getting a high quality starting material in terms of contaminants. Biologically, there are even more benefits. So these babies are 0 days old. They have long telomeres. They have healthy mitochondria. It's a really clean starting population that has massive proliferative potential as well as downstream functional potential. So I refer to cord blood as liquid gold. I think it's a great thing for any cell therapy to be using.
For sure. Yeah, it's also a good source of young MSCs
Yes,
to get them
absolutely.
injected for joint injuries and other applications. So,
Yeah.
yeah, second question, then I'll open it up to the audience. The secret sauce is this HSC expansion method you have, achieving 100,000 fold expansion. They can allow you to produce many other cell types beyond NK cells. T cells have been hot in recent years. Can you just elaborate on the advantages of NK cells versus T cells and then other cell types that you're excited about in the future?
Yeah, absolutely. So I will say that I do love T cells. T cells are not in our pipeline. That's because I think we can really uniquely able cell types that other people have been struggling with. So natural killer cells are great because they can sense virally infected cells, stressed cells, senescent cells, and potentially cancerous cells in a very heterogeneous manner. So you don't need a single antigen to target. So I think they're really great as potentially a senolytic, an infection preventer, and things like that. The reason that we then have these other cell types in our pipeline, red blood cells, mitochondria is just an organelle actually, and macrophages, is that I want to move beyond just the oncology space to the longevity space. And we're seeing a lot of really promising early advances in things like fibrosis and other areas, but with the same manufacturing challenges that have plagued natural killer cells. So I think T cells are doing just fine. That field is going to chug along on its own, but I think we really have the opportunity to help advance these other cell types into the clinic. And that's where I think we can have the biggest impact.
Cool. All right, so opening it up to questions, we have from Chris Bradley applauding the presentation here, and Greg Timblin asking about the MOA of the small molecule.
That is a secret, unfortunately. I can't say that on a webinar, but if you want to diligence us, I'd be happy to open up our data room.